Good morning. I'm Jeremy deWaard. I'm the Associate Director of Collections here at CBG. And I just want to quickly acknowledge some of the team here. It's a large team that works on natural history collection, harvesting. That's just a subset. So in the last couple of weeks, we've been thinking about or grappling with this 2.5 million bins and 20 to 25 million specimens, and where we're going to source those specimens from. And you can, I think, categorize them simply into these three categories: fresh collections, living collections, and DNA tissue repositories, biobanks, tissue banks, and natural history collections. I think the fresh collections will be, for the most part, pretty straightforward. Global Malaise Trap Program has already brought in 2 million plus specimens with not too much effort. Kruger Malaise program that we've talked about yesterday a little bit, more concentrated Malaise trapping in a particular region is another huge source of very diverse samples, and which something that Scott talked about, the intersection between Global Malaise Trap Program and Forest Geo is, I think, a really good source going forward. And I think that based on the last decade or so of sourcing freshly collected specimens, processing them, and archiving them, is fairly routine. At times when we had the funding secured, we were quite able to ramp up to almost 1 million specimens per year. And again, this year, we're up at about 700,000 specimens that, again, just here at CBG that we've sourced, processed, and archived. And so all these 4 million specimens are just sitting behind you in a couple of rooms behind Scott there. The second, living collections and biobanks. Jonathan talked about this. It's, I think, for nations that are just entering iBOL now, this might be a really good place to start, especially if you don't have a lot of vertebrates or plants barcoded yet for your nation. There's a good chance that those species could be sitting either in gardens and zoos or in a freezer somewhere. And we of course have a big biobank downstairs of over 5 million samples. And there's actually 109,000 species that don't have bins yet, sitting just downstairs. So that's 4% of the goal right there in the basement. They have failed on the first pass of barcoding, and so that's a potential source as well, just to go back with them with more modern techniques to recover bins from them. And finally, I'll talk about natural history collections now. We all know this is a huge, exceptionally important resource. There's probably somewhere around 3 billion specimens held in natural history collections around the world. They're already prepared, they're already mounted, and I think what's most important is that they're often expertly identified, if not part of the type series to define that species. There's also very rare taxa that's come up a lot in the last couple of days, and of course from restricted countries. So there's lots of pros. I think there's fewer cons. Of course, the biggest one is that you have to deal with DNA degradation if you're not working on freshly collected material. So it's a slower process, it's costlier. And if you're not in Germany, there are restrictions on access to specimens and sampling them, especially type specimens, of course. And anywhere you go, there are going to be biases in the taxa or the regions, which reflects past and present curators. So we've, at the CBG, have analyzed or sourced and processed a lot of natural history specimens, both in museums and herbaria. We have primarily done arthropods and other animals, and then plants and mosses. We haven't done a lot of work with fungi or other groups. And so we're starting to get a handle on the factors that affect the recovery of first Sanger sequenced DNA barcodes. We have a couple of papers that had a lot of specimens included. The first is Lepidoptera from the Australian National Insect Collection, 40,000 specimens analyzed there. Masha Kuzmina looked at 13,000 herbarium specimens. And you need that mass of data to tease apart the various factors. The obvious one, again, is just age, and unless you are preserving at very low temperatures into, say, liquid nitrogen directly at the time of collection, you will see this gradual degradation over time. This is from that Lepidoptera Annex project, and you can see, regardless of the size of fragment that we targeted, there's a sharp decline over time. We see this as well in other groups. This is mosses. This is unpublished from the Canadian Museum of Nature, about 2,000 specimens. In this case, and something that we've seen routinely, not just in mosses but in plants as well, that you can go quite far back in time with herbarium sheets. They're generally dried really quickly, preserved well, but it certainly does drop off at some point, often over 50 years or so. And just, of course, with other specimens, fluid preserved specimens often drop off after 5 years already due to the acidity of ethanol that they're preserved in. And other groups will show very characteristic decline with age. The marker of choice is a factor as well. Again, this is from Masha's study of Canadian plants, and you can see the variation. The variation is often associated with the marker length, or the marker length that you're targeting. And so this is a common trend that you'll see in plant barcoding papers. Collector effect is something interesting that we saw in the Annex project. You can see Stockard here, an amateur lepidopterist, and Nielsen, a professional lepidopterist, both collected at roughly the same time. But you can see here about 80% recovery on average, and Stockard's was, I think, 3% for the 800 specimens that we analyzed. So this usually relates to the method of preservation or the method of killing. We suspected in this case that it was due to chemical fumigants used in the process of killing the moths. Then, of course, you have interplay of various variables. This is from the Canadian National Collection, not just specimens that we processed personally, which I think are about 70,000, but also what CNC curators have sent to us. And so this is divided up by order and you have sequence recovery versus average specimen age. And you can see that there are certain groups that deviate from the norms. We have always struggled with beetles. We have similarly had problems with Hymenoptera. It's likely due to primer issues. We have sequencing issues as well with Hymenoptera. Coleoptera, it's probably a lot to do with the lysis of tissue, extremely hard exoskeletons, so getting at the DNA inside those little bullets can be difficult. And as well in this, if you looked at, say, the Lepidoptera, body size is a huge and important variable. We often struggle with Microlepidoptera, and that's of course just related to the tissue size that you are able to extract from, which is related to the mitochondrial DNA content in it. So we've moved recently to next generation sequencing solutions. This is a paper by Sean Prosser and colleagues, where we're piecing together 6 fragments, 6 smaller fragments, and assembling them into larger barcodes. It's a pretty interesting and complicated procedure that I won't go into, but if you can, I'll direct you to Paul's paper or to Axel's paper, where they've already looked at 3,000 types with this process. We've recently implemented it on the PacBio sequel. We're multiplexing 96 specimens at the same time. There are some issues with chimeras and pseudogenes, so validation is very important. It is a costlier procedure. It's probably talking about the $1.50 per specimen price that we heard yesterday. That 4 times was a lot higher. But we do have exceptional sequence recovery with it, even when these are the hardest samples, when it's failed for Sanger-based workflows. It's probably 5 to 20 times the Sanger-based recovery. That 20 times is probably with our ethanol preserved specimens, formalin fixed specimens. And what it actually does is it relieves you of that association with age. Plants have gone a similar way, using next generation sequencing with either target enrichment or genome skimming. We heard a bit about this yesterday with Inger, and other groups are going to full chloroplast genomes, like at the Smithsonian. And then, of course, we heard with Pete that even full genomes are being used. So this might be another area for discussion this afternoon about how this work will be coordinated and stored. Quickly, we did a pilot, again, trying to target the genera that are not on bold. We looked at 950 specimens of flies at the Smithsonian, so about 850 genera. We went in hoping that we would just get stuff from 1950 on. Average age was 38 years. We first went with Sanger, went with the targeting mini fragments, had pretty poor recovery. We've since gone back with our NGS protocol, and the recovery on those failures is about 80 to 90%. So in general, I think when you go out to harvest your natural history collection or another, it's good to have pick lists, using checklists. BOLD has a fabulous checklist system now that you can use. You probably want to set your thresholds and your expectations based on how much money you can put towards it, and all those factors that might come into play. And don't be scared about getting just small sequences. You often do get matches to freshly collected material in the Global Malaise program or other projects on BOLD. I would just, lastly, if you are interested in this, come talk to me. There's also, we did a workshop, so there's about 5 hours of talks on the different processes of harvesting from museums. And finally, Pete has led a group that looked at this after the 2015 conference. They met again in Edinburgh. And so there's a fantastic synthesis on this topic, and the link is there. And this talk with those links to all the papers is posted, so I encourage you to look at it and look at all of those talks and that synthesis. So that's it. Thank you.